Morning. All right, it's our last little vaccine update for the day. This one actually came out, I think, a couple weeks ago, and I didn't notice it. It's not a full-fledged vaccine, but this is one of the ones that was talked about earliest. It's from a company called Inovio, and the paper is called "Immunogenicity of a DNA Vaccine Candidate for COVID-19." All right, and so what this is similar to what a lot of people are doing. So they're basically taking something and using that something to deliver just the spike gene from coronavirus. They're either delivering the spike protein or the spike gene as an RNA or the spike gene as a piece of DNA that'll make an RNA that'll make the protein. So whatever. Different way to get to the same thing, but they're all trying to make spike inside of you so that you will make a response to it, so that you will make enough of an immune response to it and be protected. This is the idea. So with the DNA vaccine, they use this cool thing called a gene gun, except that they've changed the name of it. I forget what thing they're using because gene gun just sounds, Wild West and dangerous. Just like pull out my gene gun and bam, yeah, shoot you full of genes, yeah, which is pretty much how it works. The old gene guns would take a piece of DNA, wrap it around little tiny platinum or yeah, I think platinum beads were pretty common, different metal, and then just basically accelerate. It's like a little rail gun, and you sort of boom, yeah, drive them into a patch of skin. And so they'll penetrate to different depths and they'll each end up in some kind of cell, is the hope. And so then once they're inside the cell, then they're going to deliver that piece of DNA or RNA into the cell. And at that point, the RNA or DNA is going to function as instructions, just like they always do. And those cells are going to start to make whatever it says, so like a protein. A lot of people have tried this. A lot of people have tried it in a lot of different ways. It is a really good idea in that this is something that you should be able to make within a week of having a new virus sequence, and you have the new virus sequence within a day or two of the virus. So it should be just instant turnaround. The problem is it doesn't really work. It hasn't ever really worked particularly well. These are things you can sometimes get to go quite well in mice. And in other animals, the results are iffy. And so far in humans, the results are just you don't get enough of a response for reasons that aren't particularly clear because it's still a gene that's still going in, that's still being made. They scale up the dose because humans are big, yeah, but yeah, it's still not that much. So anyway, all right, how their vaccine do? So let's scroll down. It's right at the end, I think. Always is. The good stuff in the paper is usually in the last figure. Let's see. So they're putting it into mice, and the mice are making reasonable, though not high, numbers of antibodies. So normally in something like a mouse model, you want to see on the order of about, it's a dilution factor, but we'd say about 10,000, a 10,000 antibody titer or higher. And what they're seeing is about 1,000. So already we're a little on the low side for total antibodies, but as long as all those total antibodies are neutralizing antibodies, which they won't be, but as long as they are, then this still has some hope. And so we scroll down to the next figure. There it is, figure four. Figure four. Yeah. And there they have the neutralizing antibodies. And remember, our target is we want to be somewhere between the 500 might be okay. That's probably the lower limit of okay. 1,000 is more reasonable for a neutralizing antibody amount. And neutralizing antibodies are just a couple out of this entire population of antibodies that bind somewhere. So, yeah, if you can get like 10,000 regular antibody level and 1,000 neutralizing antibody level, then chances are you've got yourself a good vaccine. Anyway, I'm stalling, but yeah, they ended up with an average of about 80 and with a high of almost 150. So that was okay, but a low of about 45. It looks like they did in 5 mice. So it's a range. It's kind of low. It's a little higher than the Oxford adenovirus vaccine, which was real bad, yeah, in terms of neutralizing antibodies. Um, yeah, I don't know. it does not look particularly promising. I'm at risk of confirmation bias here because I've been around this technology enough and talked to enough people that I'm already coming into this thinking, boy, this is probably not going to work. I don't know how you'll get enough of an immune response. You can get some, but I don't know if it's going to be enough. It's confirming that right now, and when something confirms what you're already thinking, you should probably take a step back and say, just, am I still being objective? Can I find a way to be objective about this? Yeah. So right now, at least from the evidence I'm seeing in this paper, I don't think this one looks particularly promising. The most promising one that's actually in clinical trials does seem to still be the Sinovac. It gave neutralizing antibody levels of about 500. Now we need to look into these papers because the neutralizing antibody test is done slightly differently in different places, and depending how you do the test, you can get different numbers that really mean the same thing, and so it's important to know we're comparing apples to at least, a different kind of apple. Yeah. So there we go. Inovio, not great, but they're trying, and right now trying counts. Thanks very much. This has been Ask Dr. Ben.